methodology class. As we kind of mentioned this before, it's so much information, but it's just required that you have an introduction in here, and certain things are required, and that's just basically what I stick to. So it's a very, very brief introduction. So there's one thing missing off of this, and that's something about milk. But other than that, everything on here, like it's basically just saying, be able to describe this, be able to say this, know the definition of this. So it should be pretty straightforward. I have the grades up to date that don't include the exam that you got back today. Okay? These grades, you can come, whoever hasn't seen this yet, come to me in lab and I'll show it to you. This is based on 45 out of 50 technique points, which is about like the max that people get, and 50 points on your lab notebook, which I truly believe that there's no reason why everybody wouldn't get 50 points on their lab notebook. If you just didn't follow that sheet that you downloaded that said how to do it, if you really just like didn't follow that at all, then you probably won't. If you missed a lot, then you probably won't. If you don't write up the very last labs, then I'm going to take off a few points for that. If you consistently don't ever include a discussion, or just something like really obvious like that. Otherwise, I do not go through and nitpick the lab notebook. And I kind of mentioned this before, but now that you've been working on the lab notebook, this will probably make more sense to you now. What I do is I go through, I make sure you have a table of contents and you number the pages, just because I asked for that. Again, if you follow that sheet and follow what I said on that sheet that you can download off the website, it said, start like that. Then you have an objective. And the objective is just like, what are you getting out of the lab for that day? It's a study of, pH ranges or whatever it is, just something simple. Then you have all of the information about the lab, how you're setting it up, what is the preliminary kind of information you need to know, what are definitions or whatever, what are results, and then a discussion. In the very beginning, the discussions were like really not really necessary, because in the very beginning we were just looking at different organisms from different groups. But then after that, you started doing actual experiments where in a discussion you could point out what went right, what went wrong, what are the possible reasons why things went wrong, what could make it better. Like if you did something with gram staining, you would talk about even making sure that your slide was evenly, thin, and that you were more careful when you made it from a solid, and then you put extra drops if you made it from whatever helped you. So that's like basically it. So when I go to grade it, I look for those first things and make sure that you're sticking reasonably to a standard format. Then I choose a few labs. Like right now, I just said gram staining. Yes, I'm going to read the gram staining lab. I'm going to read the population counts lab. I'm going to go through and pick out a few labs that I know that there's plenty of things for you to say in the discussion. Not that the discussion has to be really long, but that you actually put some thought into why things worked well or didn't work well. The pure culture lab. But especially something where you actually had a chance to really get comfortable in the lab and get comfortable doing the lab work. If those look good, if you have food, water, and milk nicely written up with a good discussion, then I'm pretty much done with your lab book. I'm grading your lab book like this. Okay, I do not go through and read everything in there. That would take me forever. It's worth only 50 points. And if it's done really well, it's only going to benefit you on the lab final. So that's how I approach it. I just think like it's not necessary for every single lab to be perfect. But the better it is, the better it helps you. And as long as you basically respected what I asked you to do, then I'm going to give you some leeway. Yeah, you did better on these labs. These labs weren't as good. But for the most part, your lab book was good, 50 out of 50. That's how I do it. So that you kind of know, like, don't waste time and go through and try to make everything perfect because you think I'm gonna read it. Try to make it perfect because you can use that information. If you put too much information in there even, like, it's not gonna help you in the lab final either, because then you're just like sorting through a bunch of writing to try to find the answers. So doing a lab notebook is really to help you guys organize your stuff so that you can study from your lab notebook and not have to study from a whole bunch of different sources, like the lab book, plus whatever I'm talking about, plus where your results are, and stuff where you have everything in a separate place. If you have it all nicely in your lab notebook, that's better. If you did your lab notebook on the computer, I'm gonna remind you again, don't take it and cut it out and glue it in a lab notebook. Print it out and staple it together and give it to me. That's perfectly fine. You know, some people are just comfortable with doing all that on the computer. The problem with doing it like that means you're not like writing it as you're doing it, so you still have to like go home and do it, but don't waste time like gluing something to a pages. That's not necessary. If you're really behind on your lab notebook, finishing it after the lab final, I would have to question whether or not it's worth the time to do that. Like, how much are you really missing? How much would I really take off for that? And how much study time would you be missing out if you spent all that time after the lab final? So otherwise, I'm expecting that you will turn it in at the end of the lab final. How the lab final works again is that you're going to take the test. When you finish it, you're gonna raise your hand, let me know, I'm gonna give you 10 minutes. And then at that point, you can look up anything, so if you've like really organized yourself and made notes off to the side and circled things that you have to go back, then go through and use that time wisely. At the end of that time, you close it and give it back to me, everything. The final plus the lab notebook. So it's not like you can use it in the middle and try to memorize everything at that point and, you know, then close it and then finish it. No, when you're done with the 10 minutes, which really turns out to be probably more like 12 minutes, because I give you the benefit of like the minute that you raised your hand and got it out and got yourself settled to the minute that's over, you know, like that. So, you know, you really have like the full 10 minutes at least. But don't rely on 10 minutes being that helpful, because 10 minutes goes by really fast. You can't look up everything in 10 minutes. So what you really want to do is make sure that you have studied well for the lab final, because that one you don't get to drop. And I have the grades. I think like probably a lot of you are probably doing maybe even a little bit better than you think you are. Once I have everything in, so I have all the quizzes in here, I have the unknowns in here. It doesn't have any extra credit from the computer center, because I won't get a printout of everything until probably sometime next week, the end of next week, or even finals week. So you can add that in there, and then you can add in the exam from your previous exam. If you add that in, then you're basically almost assuming that you're going to drop the lecture final. So you can figure out like what it might be if you got a better grade on your lecture final and dropped one of the scores. So you'll have to figure that out for yourself. Okay, so 4 o'clock tomorrow. I'll stay as long as people want to go over the lab stuff. So any questions about any of that? Yes.
How long did you say you would be there for?
I'll be there as long as everybody wants to be there. I'll get there at 4, and if you guys are there for like, you know, I'm sure it'll be like, I would guess around 2 hours. How many people could make that? Maybe we need it to be later.
Yeah,
Yeah, I'm sure that we'll still be here at 5. There's no, I mean, I can't imagine that we would get done. I've done this kind of thing before, and it usually is around a few hours. Yes.
Where is it?
In the lab. Yes.
For the final, do we know like all the indicators for like all the 20 tests that we used for unknowns? Like,
All the indicators that are like in the media.
yeah. Okay.
But you guys know that already, right?
Yeah.
I would think you would know that already. And it's not like I would be saying like there's only 100 points. You know, so if you look back at all of our labs and think that I'm gonna ask a few questions from every lab, and then it's gonna be weighted more heavily on food, water, milk at the end, then how many could I ask you about? I can't imagine that it could be without phenol red. Everybody should know phenol red, it was in so many things. And of the other indicators, methyl red, you added to it. Bronchithiol blue was really the only other real indicator, wasn't there? Was there another one? Any intermediates? So that's really like 2, right?
Yeah.
It always sounds more overwhelming. But you should know the reagents and stuff like
For this, are you having set up like stations or is
that.
that?
No, it's just gonna be an exam, and you do need a scan. It's like a giant.
It's a same
lab
form?
quiz.
Is there an essay
It
portion?
really is like, yes. Remember when we were discussing things and I said, oh, this would be like a good question that I always ask, who remembers what I said? What do I always regularly ask what I should say? Why do this test if we're already doing these tests? Why do a heterotropic plate count if we're already doing an MPN or a membrane filtration? Advantages and disadvantages of the multi-test systems, right? The miniaturized multi-test systems. Advantages and disadvantages of like membrane filtration versus the most probable number. Those would be things that I would be expecting, even if I don't specifically just ask them. And if you go back and you look at the stuff that I've been telling you kind of to do over the past month, you know, go back and look at your protists. What were the most important protists? What are ones that you know I'm going to ask? Do you think I'm not going to ask about Plasmodium vivax? Do you think I'm not going to ask, like, what's a prokaryote and what's a eukaryote? Since so many of you, for some reason, missed that on the quiz. The only prokaryotes are bacteria. Right? None of the fungi or protists or helminths are prokaryotes. Okay, like you know I'm going to ask about malaria. You know I'm going to ask about Taenia solium or Enterobius vermicularis or something, because I'm asking myself, what do I want to make sure my students know when they get out of this class? All those things that are going to be part of like your life. That's going to be number one. Then after that, I'm going to be looking at, some more challenging questions because I want to see like who's the A student, who's the B student, who's the C student. Know like who put the effort in to really know the material. But I want you to know like the kind of stuff that affects your life. And understanding about water testing and food testing and milk testing and how to make determinations based on what you know, I think that's really important. I mean, honestly, I don't know how people live their life without knowing a lot of this information. It's so important in everyday life. I just don't see how, like, if you never knew any of this, you would be like, you know, I don't want to use the word ignorant, but like you would just be unaware of, you know, things that make people sick and stuff like this. In this day and age, I just don't see how people could be like that. or why they wouldn't want to know this.
Do we need to know any of the life cycles?
No. Because life cycles were only in lecture. We never learned the life cycles on lab. So you go through, you look at that stuff, and you'll get it. Plus, knowing the stuff that you're learning for lab right now is also like really overlapping in lecture. So right now, I'm going to talk about the water and food, and hopefully that will help with that too.
Would studying or quizzes be a good study guide?
I mean, there could be a couple questions that come from the quizzes. Just because the quizzes in general will have more detail on them because there are a lot of questions over just a few labs at a time. But I mean, I wouldn't be surprised. You know what one I always end up putting in there is something about the measurement of a zone of inhibition. Just because a simple question like that shows me that you knew something about that lab, that you had to look it up to see whether or not things were intermediate, sensitive, or resistant. That's a good all-around question because it shows me that you understood what you were doing in that lab. And also it helps you understand that some bacteria are resistant to the antibiotics, some are sensitive to the antibiotics, and some are kind of in between. They're, intermediate. So like that one little question answers a lot, and that might have been on a quiz before. So there could be something like that, like some of my favorite questions.
What's the format? Just like quizzes?
It's like our test, basically. Half of it will be on a scan prompt, half will be on
Jericho's testing.
You're talking about. She has earphones in. So everybody can hear you. Everybody can hear you. And you're just breaking lights. Okay, what
Good,
else?
good job.
Ask it.
Ask.
It might be
Anybody
helpful.
else
Ah.
want to know?
Like, I'm really bad at conversions, and I know that was at the beginning of the class, but this has something that we should, like,
Oh my god.
Oh, I will, I definitely will ask you to understand about measuring organisms, but not to
Oh.
calibrate an optical. So you never really had to convert,
just like-
you won't
like
have to convert. And for sure, you
Okay.
know you have a dilution problem, at least one dilution problem. But understanding the concept behind that is important.
This is the fourth one screen.
See, everybody appreciates.
Definitely.
Anything else? Anything? Did you guys all like read what you were supposed to do in lab tonight? I wonder how many people actually downloaded those things. One person.
downloaded.
Pardon? It doesn't have the immunology. That's the next one? Oh, okay, for the next one then. Oh, that's good.
We're all together. Right?
Okay, did somebody else have that? Okay, so all of this stuff is gonna help you
Oh, have you
hopefully
studied?
with lab 2. But basically, in this particular chapter, if you look at this study guide, which you can download, it says, number one, the concerns with water quality. And then I have listed resistant organisms, low-dose organisms, and organisms that are not killed off during water treatment process. Then you want to know about indicator organisms, that's what I'm going to talk about tonight too. And then testing for water quality, which again, like just is what you just did in lab. So there is some additional information in here, just in general discussing what happens in water treatment. So I left it in there because I'd like you to have an opportunity to see that, but I'm not going to ask you about water treatment for the fungal. Okay, but I do want you to just be aware of it. So it's important because we're really looking at pathogens that can survive in water and that are a health risk. And we really divide those up into those three groups. The organisms that are resistant to any kind of treatment, organisms that are low-dose organisms. Okay, a low-dose organism meaning that even during treatment, if every single last one wasn't killed off, you would have a low dose there that might still cause disease. So organisms that are resistant, that really overlaps, of course, with the organisms that are not killed off. Okay, so that's not on this slide, but those are the three things that we are really concerned about in water.
So, can you say those again?
The low-dose organisms, organisms that are not killed off in water treatment, and resistant organisms. One thing that you're probably not really thinking about at this moment, but we kind of brought this up before, and that's that there is an overlap or an influence on antibiotic-resistant organisms and organisms that are going to be resistant to disinfectants. So we kind of mentioned that the whole thing of using hand sanitizers and all of that all contribute to antibiotic resistance because some of the same mechanisms that help protect the organism against some type of disinfectant or antiseptic could be the same mechanisms that prevent it from being sensitive to particular antibiotics, like the ability to push that chemical back out of the cell, those multi-drug pumps they're called. So sometimes that is going to really be an overlap. So both things really influence each other. Having a lot of disinfectant in our water contributes to antibiotic resistance and vice versa. There are other reasons why some organisms might be resistant. Right? What other reasons might there be that organisms might be resistant to treatment or anything? Right? Like we could have cysts, protist cyst. We could have biofilms. Did you just say that?
No.
Oh. What did you say?
I was gonna say like if they have capsules or spores.
Right, if they write an extensive capsule, right, the extensive glycocalyx, that's an important one. Especially those organisms in water like that. So that's a good thought. Thanks for, I wasn't even gonna mention that. Organisms that are like in clumps, like colonies, or when there's extra organic material there, so that they're sort of protected, like the idea of what happens in a spa. We kind of talked about that before. You have like sloughing off of skin cells, so you have all those like extra organic material. So even though you might have a lot of chlorine in your spa, if there are a lot of people in the spa, and like literally you slough off millions of cells, skin cells, and then the temperature is not going to be the right temperature. You have like, evaporation. So that's why like in spas, chlorine doesn't really work as well like that. But just that extra organic material, remember in the last chapter, extraneous organic material, very important, right? Because it combined with chemicals making them less effective. So all those things are going to be concerns. Okay? The organism itself, plus those certain conditions that lead to them being resistant. And then low-dose organisms means that you have to make sure you kill off every single one, because if only one is the dosage that's going to make you sick, how likely is it that every single one is going to get killed off? So this table shows basically some of the organisms that are listed. This is the water contaminants listed by the EPA. And if you look at these are all things pretty much that we talked about. The norovirus, that was one that you had to learn about when we talk about viruses. It's just in this Ehrlichia viruses as a category. Campylobacter jejuni, that was a concern because it is a low-dose organism. Entamoeba histolytica, same thing, a low-dose protist. E. coli 0157H7 can be low dose, but also just because of its severity. Helicobacter pylori, you learned about that one, right, that caused peptic ulcers. Hepatitis A, we didn't really talk much about hepatitis A, but hepatitis A was listed when we talked about viruses, and that's one in which it's pretty easy to pick it up. You know, you can pick it up from contaminated food and such. The problem with viruses in water is that viruses can remain effective even if they're exposed to chlorine, so it can stay infected for a long time. Legionella pneumophila, that was the one that's a concern because you could inhale it, the one that's associated with Legionnaire's disease. Naegleria fowleri, remember that one? The one that you could get, you know, like up in your sinuses, like usually like water gets up your nose. Salmonella, Shigella, and Vibrio. So all of these are all organisms that now, because of your last exam, you had to learn about all of these. I don't know how you guys will feel about this, but I know that I will regularly get emails or little texts from students that are in the nursing program or working, that they're just like, oh, they're so like amazed because now they're in the nursing program, and every time these things come up, it's like, I know that organism, I know about that, oh, I've seen that test or something. So it'll be nice for you guys. Don't forget about all this information. So this is really just examples, and basically I put these examples in those three categories, right? Resistant organisms, or organisms that are not killed off, and low-dose organisms. Okay, because they all kind of just fall into those categories. Again, looking at water purification or water treatment, you can see basically what happens. It tells you what the process is. When our semester was longer, we used to regularly take a field trip to the water treatment plant. You could go in their lab and see how they're doing their testing and see exactly like basically what you guys are doing, and you have an opportunity just to see what they do to treat it. But when they shorten the semester, we just don't have enough time to do that. But when you look here, and you have this in here, it just kind of shows the different stages. They're not necessarily all in like some kind of container like this, but it shows like what happens, that you have precipitation, and then you have removal of that. They add a flocculating agent, which is like some sticky stuff that makes everything like clump together, and then they let that settle out, and then they filter it, and then they go through an aeration process to try to like help get out like the smells or anything that's associated with it, and then disinfection. If it's disinfected with something like ozone, which prevents development of some of those carcinogens, then they still have to add chlorine after that. Right now, your water does have to have a free residual chlorine so that it remains safe from the time that it leaves the water treatment plant till it gets to your house. So I won't be asking you specifically about this, but it does help you kind of understand the big picture. And then, again, this is just kind of bringing up those same examples of the organisms that we're concerned about. The only thing here that is added in here, Cryptosporidium, Giardia, of course, you know, besides just the Entomema Histolytica. So really, we're looking at Protus, and we're looking at things that like Formosus like that, and then those bacteria that are also low-dose viruses, also because they are not necessarily made ineffective by. Usually with viruses, they're going to be filtered. When we are going to analyze the water, that's basically what we're doing in lab right now. It's based on detecting indicator organisms. And this is really important. So this is your first, your essay type questions. It's extremely important for you to understand about indicator organisms. I've mentioned that, I've thrown that out. We talked about coliforms. I've said all this, I've given you the definition of coliforms, but here's where it ties all together. In public health, you cannot test for everything. It would be nice if all of your water was tested for Giardia and Entomema histolytica and Cryptosporidium and all the different viruses and E. coli O157H7 and, you know, Shigella. And how could they do that? They can't possibly do that. Right? But what do all those organisms have in common? Maybe except for like Legionella, and that's, you know, kind of uncertain about where it originally came from. But they, for the most part, all are carried in feces. So the idea, again, is that if there was a way to detect whether there's been fecal contamination, then we think there's a really good chance that something is going to be in there. When you start taking hundreds and hundreds of people that are contributing, thousands of people, millions of people in a metropolitan area, all contributing feces into the sewage system that ends up, you know, in water, or you have all the people that are using most of where our water comes from as a recreational area. When you think of where our water comes from, most of our water comes from open areas, right? It comes through the aqueduct, it comes from lakes, it comes from Cascade. Things that are open for not only just people to be in there, but animals, birds flying over it. So we know it's all contaminated to begin with. Some comes from groundwater. Groundwater is usually going to be not as contaminated. But I say not as contaminated because there's other ways that sewage gets disposed of in rural areas, and we have plenty of rural areas around us. They go in septic tanks, and they have leach fields in which it all goes out, and then it like percolates through the soil. And if things are not built to the proper specifications, then you get contamination of feces from septic tanks. Septic tanks leak, they back up. Leach fields get backed up. When people drill water wells, like in rural areas, there's all kinds of guidelines about how far away they have to be from all of these, but I'm sure that you can understand that things don't always happen exactly that way. Right? Like you all know people, I'm sure, that have well water for water, right? You all know somebody that's lived in Agua Dulce or Acton, right? Nobody in here knows anybody that's ever lived there before.
Well, I used to have to drink out of well water when I was younger.
It's not uncommon
in the Philippines.
in our area for people to be using well water.
I was in a different country though.
But yeah, Agua Dulce is all well water.
Really?
Nobody knows anybody that lives in Agua
Good
Dulce.
time to go there.
Okay.
And all kinds of communities like have well water. But at least those are in, you know, areas that you would expect them to follow standards. Otherwise, like you know there are times, especially in rural areas and or rural areas in other states and such, that nobody is coming out and inspecting every single well and making sure that everything is exactly to specs. So sometimes underground water gets contaminated like that too, and from mammals that live around that dig and such. So when we're looking for an ideal indicator, then we're looking for an indicator that's gonna really be able to show us, like, we want to know all the time that if we're looking for an indicator, that it is actually showing us what we're looking for, which is fecal contamination in this case. Indicator organisms could be used for other things as well. They're indicating contamination simply because we can't test for everything. You guys are getting that, made that concept, right? Okay, so what is an ideal indicator then? It's suitable for analysis of all types of water. So if we have like the perfect indicator, then that means it doesn't matter like what the water source is. It's gonna work for all of them. It's present whenever the pathogens are present. Okay, we don't want to find an indicator, but there's no pathogens there. That would be called a false negative. I mean, a false positive, sorry. A false positive, because if we find out that we think there's pathogens there, but there aren't, that's just then costing us money, because now we have to go and treat that. Where do you think the money comes from? Like your taxes, right? And every time that there are tax cuts or anything, like, do you want to vote on these things and decide to take money away from this kind of stuff? So like we want to use the money wisely, because we don't want to not have these particular types of testing done. That's what happens in other countries and why you can't really trust drinking the water in a lot of other places. So we want to just find it when they're present. We don't want to find it when they're not present. We don't want a false positive, but we don't want a false negative either. Survives longer than the hardiest pathogen. Right now, this is what we don't have. If our indicator organism is a coliform, which is essentially E. coli, okay, we know it doesn't survive longer than the hardiest pathogen. We know that for sure. Okay, so right now, that's the problem. Okay, and that's a big problem, because that means we do get false negatives. We test, we don't find any coliforms, so then we think, oh, everything is perfectly fine because there were no coliforms. Okay, and why do you think that's the case? Why am I saying like 100% sure that we know that that's not the case? Because what is still there when E. coli has died off? Protists, cysts, viruses. I mean, spore-forming organisms is just that there aren't any spore-forming. Like if you ingested a spore, it's not going to cause a problem. Okay, like it's only when spores get in something like an anaerobic condition. Okay, like right now, there aren't any spore-forming organisms that we have concern about. Unless they get in an anaerobic condition. So in water, that's not a concern. On a surface, that's not a concern. It's only if they get into something like canned food. Okay, so protists, for sure, and viruses, they can both remain viable long after E. coli or coliforms have died off. That's a problem. Does not reproduce in contaminated water. We don't want them reproducing. We don't want to add to extra organisms being there, but also we want to be able to have an idea how much contamination is there. So if we have the organism reproducing in the water, then we just end up having a false idea of how much is there. Again, we're talking about something ideal. We want it to be able to be detected in a specific test where we're just getting that particular organism So something that's easy and something that's sensitive so that we are just getting the coliform, that we're not finding all these other organisms and thinking they're coliforms. Does that make sense? Harmless, of course, we don't want it to be something that's harmful. Its level and water reflects the degree of fecal pollution. So that kind of really overlaps in the does not reproduce, because we want to have that level just tell us that, oh, there's a lot of contamination here, or oh, there's a little bit of contamination, because that's going to help determine what kind of treatment has to be done in order to get it to be safe water. So I for sure will ask you to list at least some of these, if not all of them, but I hope you understand the big picture. Normally, I ask about like, what is an ideal indicator? Why do we use indicators? Why are indicator organisms important in public health? And
Didn't you say that E. coli is an indicator organism? But it's not like harmless.
regular E. coli is, like E. coli that's just happily living in our intestines. I mean, if it gets someplace it's not supposed to be. Otherwise, that's why, like, in lab, you can use E. coli and not be, like, fearful that it's gonna cause a problem. But if it was a particular strain, or if it ended up someplace, like E. coli is a common cause of urinary tract infections, because if it gets into the urinary tract, then it can cause infections, or if it gets other places. Okay, so any questions about that? So you know there's going to be some kind of essay type question about indicator organisms, the importance in public health, and some of these, ideal indicator, characteristics. The ones that we're familiar with, coliforms for sure, and you know that definition of the coliform. Understanding a little bit about that definition of the coliform does help you understand water testing in general. It helps you understand what we're doing in lab and what that actually means. So, I'm not going to ask you to just write out that definition, but it is helpful if you know that definition. Remember that definition, that it was a rod-shaped, non-spore former, fermented lactose with the presence of gas in 24 to 48 hours. Actually, it's like a facultative endosome on top of that. Have the whole definition. Brand name. Fecal streptococci, or the enterococci, that was mentioned last time around. You had to know that for the last exam. And those, it says, increasingly used to test brackish and marine water. So cold water, also frozen foods, that was mentioned as well. Just because it survives a little better in those kinds of conditions, colder water, saltier water, and such, than E. coli. Okay. There are more and more other things that are being looked into. One of them was the coliphage, the bacteriophage that are specific for E. coli. And I mentioned that before too, because that virus will be around because the E. coli is around, but it's still going to be there after the E. coli dies off. It's still there. So if it's still there, it means that E. coli had been. So here's that definition: facultative anaerobic, gram-negative, non-spore forming rod-shaped bacteria, ferments lactose with gas within 48 hours. So you can know that basically that's kind of what we looked for. We put the water sample in lactose broth. It has the Durham tube to capture the gas. So it's going to show us if it fermented lactose and produce gas. That's part of it. That's gonna give us a presumptive positive if it did. Then we have to continue to go on to find out, is it a gram-negative, is it non-spore-forming rod-shaped bacteria, just to verify that whatever organism in fact did ferment that lactose and produce gas really is the indicator that we're looking for. So this, I know it's kind of a little, I know you probably can't read the little writing, but if you look at this test, see here's the 5 tubes at each one. Okay, we use 3 tubes, and we're gonna look at the statistical chart that's based on 3 tubes rather than 5, but this is what's normally used. So the first 3 we put in 10 mLs, the second 3 we put in 1 ml, the third 3 we put in 0.1 ml. When you guys go into lab, you are gonna count how many of the 3 had positives, how many of this 3 had positives, and how many of this 3 had positives. How can we tell whether they're positive? They have gas. And I know Kelly Burke had brought this up, that she asked her students, why aren't we using phenol red lactose broth? Why are we just using lactose broth? Because in our definition, we don't need to know whether it produced acid or not. We are simply looking to see whether gas is produced. Okay, so a positive, gas is produced. If gas isn't produced, it doesn't matter how much organism is growing in there. If it didn't produce gas, it's called a negative. So this is something that, again, when we go to lab, I don't need to talk to you about this because now you know what you need to do. I'll have the chart at the front table. You're gonna look up if all 3 were positive, but none were positive on these 2, you look up 3, 0, 0. And then it will tell you, I showed that to you last time, I showed you the chart and showed you how you would read how many coliforms per 100 mLs. So today, when you guys go to read all your results, the one thing that is going to get you all is how to write your results properly. Okay, for the most probable number, you're going to look up what it means, okay, in that chart. And it's per 100 ml. So if you remember last time, I said that you would all be responsible for recording results from all the samples. So we'll write them up on the board. And we'll just go round and round trying to get you guys to write them up properly. When you read your results from the most probable number, you will write them up, you know, X number of coliforms per 100 mLs. Where do you get the X? By looking it up, because the most probable number is a statistical estimation. Okay. If you have 0, 0, 0, okay, so none in the 10 mLs, none in the 1 mLs, and none in the 0.1 mLs. That's 0, 0, 0. That means when you look it up in that chart, because again, you have to even look that up. Because can you really just say, because you didn't have anything growing in there, can you just say, oh, well, this water is perfectly fine then? Why?
There's probably like,
I can't really quite hear you though.
The ones that are particularly before would cause gas, and we know where it is, and it wouldn't cause that reaction.
Right, that's one thing, right, exactly. And you're only testing 33.3, or in this one, 55.5. So you really can't say, oh, that just means everything is perfectly fine. So when you look up 0, 0, and 0, it's going to tell you that's less than 30 coliforms per 100 mLs. Okay? It's not going to say that means there's nothing. It's going to say statistically that means there's less than 3 per 100 mLs. Okay? And why is it important that we even say it all the way to this point? because you need to have that understanding of what this kind of testing means. We don't want to interpret something that is not really there. Like for instance, if you went through a whole dilution process on your food and milk, and then you looked at a plate and nothing was growing on it, does that mean that that food is sterile? No, it just means that either you over diluted it, or you might not be in the right media, or maybe there really isn't any, but you don't really know that. You can't make one result like that mean something more than it really is. So if you diluted it especially, then we know that it has less than a certain number. We just can't say it has 0. Maybe everything didn't grow. Maybe it didn't incubate long enough. Maybe the colonies are too small to seed. These are kinds of things that by now I could expect you, like on the lab exam, to be able to think of some of these things. Right? Like when you go in there, you're going to be able to see like, oh wait, maybe I really didn't mix up the sample with the agar that well. Because like I said, you'll see like a swirl of growth. And then you'll see all these teeny tiny little colonies. And here's something else that you guys haven't seen before. What do you think happens when you mix up something that has a lot of organisms in it inside agar? Do they all like swim up to the top and grow right on the top of the agar? So you haven't had to see that before. No, they will grow inside the agar as well. But when they're inside the agar, they can't really spread out and get really a big colony, right? Because they're being kind of compressed by the hardness of the agar. So you see like all kinds of little dots inside the agar. Can you just ignore those like they don't exist? Right, because like if you think back to what it means when you have anything growing in the agar, what does it mean? It means that it came from a single organism. If you were lucky and you really got everything mixed up and you had the right dilution, then every single bacteria actually grew like it was supposed to. Then each one would grow into a colony, right? One colony would be approximately equal to one colony forming unit, one organism. They're called colony forming units, remember, because we know it doesn't always happen like that, right? What if you had 2 bacteria what if it was a diplococci and they only developed into one colony? Right? What if it was a tetrad? So there really were 4, but they only developed into one colony. I've asked on the exam a true false, like, well, since some colonies are really big, we count those as 2 because we just assume that they really came from more organisms. Would you say that's true or false? How many people say true? How many people say false? Right, that would be false, right? Because the assumption is one single organism would develop into a colony. If the colony was big, it usually just means that either that organism grows like that or it didn't have a lot of competition there, so it could grow bigger. So if it's in the agar, it's treated just like anything else. So it's a little challenging to count them all, but you do have to count them all. Okay, so, and this one back to this particular test, but just remember as you're learning this all along, this one, you have to look it up and you have to follow the number of coliforms per 100 mLs. And the least number you could have is less than 3 coliforms. And sometimes it might be, like if you have everything growing, it might say it's greater than a certain number. Okay? If we had more time, we would take whatever is positive here, and then we would add it into another media. So down here, it says like negatives and positives. You can't really read that, but the positives then go in brilliant lean lactose bile broth. Okay, harsh media. So we have those organisms that might be injured. Now they are back to life, growing happily in this tube, and now we can put them on a harsh media. Okay, so That's the confirmed test. Presumptive, we look it up, confirm, we're just confirming that it is growing, even in that harsh media, then that means that's most likely a gram-negative rod that is, because we're putting it in something that's gonna be selective for those enteric organisms, something that has bile in it. And then once we get a positive there, we're gonna put it onto EME or endo-auger. We're gonna put it on a slant, a TSA slant, grow it, check to make sure that it's a non-spore forming gram-negative rod. All of that, all of those stages are all included in the most probable number, the multiple tube method for most probable number. This has been countless times on the lab exam. And I always say, what's not included in the most probable number? And one of the things I say is gram staining, and then you go on to do the MVIC tests just to try to make sure that it really is E. coli over something else. Looking at what a spore stain, all of that is included in here. Okay, the one thing on the question that I normally use is membrane filtration, and everybody chooses gram staining and spore staining and such over membrane filtration. Membrane filtration is a whole nother test. It's not included in the most probable number, but all of these things are included in the most probable number. Okay. Presumptive, confirmed, completed. So it's a 3-part test. By the time you actually really find this out, it's been at least 3 days, if not 6 days. Okay, that's kind of a problem then, because like you drank that 6 days ago. There's no place they can store water for that many days and keep it still clean for you. So yes, people have already drank that. That's why you get positives and you look at results already at the presumptive stage, because you don't want to wait all the way till here. Okay, so that's where membrane filtration comes in, because membrane filtration is going to be much quicker. Membrane filtration is going to be much quicker because that you can get done within 24 hours. There's some advantages, and you can see there are some other tests that they look at too. And I mentioned some other ones in lab too. But in membrane filtration, this kind of gives like some of the advantages and disadvantages. That you can filter larger volumes of water. You can get you can save time, which is really important. They say lower cost in comparison, even though I think that's kind of questionable, but in our case, it's not a lower cost, but when you reuse the equipment and all you change in between is just the filter, because those filter units that we use are really expensive, but they can just change the membrane filters so they are not using a whole complete thing. So like for our lab, it's not cheaper, but in the real world it would be cheaper. Reproducible, because you would regularly get the same counts. What a disadvantage is you couldn't use like something that was really turbid. If you had a lot of bacteria, it would interfere with the results and other things like contaminations. But a big disadvantage that's not listed here is you're putting the water directly in a harsh media. And when you do that, you're much, much, much more likely to get false negatives. If you take viable but not culturable organisms and put them directly in a harsh media, then you're much more likely to get the organism not growing. Okay, so they do have certain injury recovery techniques, like putting the soft overlay on it, putting the filter on that and letting it just like make its way down to the harsh part. You could get 50% more. We did this in my lab when I had a public health micro lab, and you could get 50% more regularly by doing that. 50% more is a lot more. also something was mentioned in here too about fecal coliforms, and I mentioned this one time before, but I want to make sure that you